Hello and welcome to this presentation on the introduction and overview on the Thermo Scientific HDX LC-MS workflow. My name is Kai Scheffler. The HDX workflow allows for protein structure elucidation and confirmation and is based on the identification of sequence sections of a protein that are exposed on the surface of a three-dimensional structure. The obtained results can be used for the assessment of protein folding, conformational changes, protein-ligand interaction studies, epitope mapping, as well as for the analysis of biosimilars. The concept of the workflow is to take a clean protein, expose it to D2O, also called heavy water, to exchange all the surface accessible hydrogen atoms versus deuterium. And that's been done at various time points. Then the reaction is quenched and the protein is digested by run through an immobilized enzyme column, typically using pepsin. The resulting peptides are then separated using chromatography detected with mass spec, and then the obtained isotope patterns are indicating the degree of deuterium uptake when we review the different time points. The change of the isotope pattern is based on the different mass of a deuterium atom compared to a hydrogen atom. The workflow consists of 4 steps that are shown here. The first one is relating to the sample preparation that entails the labeling steps, so the exposure to the D2O, and also the inline digest using the immobilized enzyme. The resulting peptides are then separated using chromatography. MS data are acquired on a mass spectrometer, and then sophisticated software is required for the data analysis. There are a number of experimental challenges involved in this workflow. First of all, the protein of interest that's used for the study has to be in a rather high concentration because it's diluted in D2O for the exchange of hydrogen for deuterium. It also has to be in its native form, so that means no acids or organic solvents can be involved because that would disrupt the three-dimensional structure. The exchange of hydrogen versus deuterium, it's also called the labeling reaction, needs to be really precisely timed because the shortest labeling times are typically just a few seconds. The quenching step will stop the labeling and minimizes the back exchange when the protein is kept at low temperature. The inline digestion is required to be performed at low temperature and also under the acidic conditions that's provided by the quenching step. Fast chromatographic separation is required also to prevent back exchange, so one needs to have fast chromatography. It also has to be reproducible. It has to be performed at low temperature and also low pH. For the mass spectrometer, high resolution is really crucial because it's needed to detect the isotope patterns of larger peptides, higher charged peptides, and also for distinguishing overlapping patterns for peptides that are close in mass. Also, it's necessary to get the complete sequence coverage of the protein, or as complete as possible, for accurate measurement of the deuterium uptake. Lastly, the applied software for data analysis needs to be capable to determine the precise level of deuteration. So this quite extensive list of requirements is in every single point fulfilled with this Thermo Scientific HDX LC-MS workflow. This photo shows just an example of a setup that we had once in the marketing lab in San Jose, consisting of the HDX autosampler here in the middle. Here in the back we see the pump and also the Orbitrap fusion mass spectrometer. Here you see the connection between the column that's located here in the box. We'll review that in more detail in a couple of slides. And that's hooked up online to the source of the mass spectrometer. Looks like a thick white connection, but it's actually a peak tubing wrapped in bubble wrap for temperature control, also to prevent the back exchange. Here, this overview shows the same workflow as we've seen just on slide number 4, with a bit more specific details relating to the individual modules involved in this setup. Here on the left, we have the HDX autosampler that's controlled by a software called Chronos. It's also provided by Leap, and the software also controls the LC and DMS. It's a fully integrated software solution. Here we have then the ultimate NCS 3500 RS pump, followed by 2 examples of mass spectrometers representing the Tribrid and the Q-Exactor family, and lastly 2 icons of software for data analysis. So now let's review these different modules in more detail, starting with the autosampler. All sample handling to create the labeling time curve is performed with the Leap HDX autosampler. It has a single head. It's using syringe exchange for different syringes, one of which is actually cooled. The sample vials are provided in these 2 separated trays. They are Peltier cooled for keeping really stable temperature. Also, we see a separated standalone column chamber for the pepsin column on the very right side here of this white box. And inside this square box, we have a flexible 3-valve setup and also the trapping and the analytical column for the peptide separation. 2 screenshots of the Leap Chronos software are shown here. Again, the software fully integrates the LC and DMS, meaning the Excalibur software. It also provides full editing capabilities for method customization. And here in the smaller screen capture, we see the time scheduling of the individual time points of the labeling reaction, indicated with these red horizontal bars. All this scheduling is automatically done by the software. The user just has to define the time points, how long the labeling reaction should be, but the scheduling itself is all done by the software. So the entire hardware and integrated software solution ensures a very precise labeling condition and also the scheduling control. Due to really good contacts and collaboration with Leap, and that's thanks to Terry, we have immediate access to all the latest Leap technologies, firmware, and software updates and improvements. And that's a big differentiator compared to our competitors. Now let's have a look at what's happening inside the cooling chamber that has the flexible 3-valve setup. And let's start here with the configuration on the left side. So first of all, we need to inject the protein sample that's loaded into the sample loop, and then run via the pepsin column to perform an inline digest while it's flowing through. So at the end of the pepsin column, the protein is chopped up into peptides, and they're further transported onwards onto the trapping column. Anything that's not binding to the trap column is running through the waste. The flow is provided by the loading pump that transports the sample by the pepsin column over the trap column. And in parallel, we have flow from the gradient pump, high pressure gradient pump, that equilibrates the analytical column that's currently not on the flow path. Now in the next step, we do switch the valve in the middle that takes then the pepsin column out of the flow. The flow still provided by the loading pump then runs via the trapping column out to the waste and cleans everything that's not binding, any residual salt and things that we don't want to push later on onto the analytical column. The equilibration step on the analytical column is still ongoing. And then in the next step, the center valve is actually switched back, and also the lower valve is switched once. So now we have the pepsin column back in the loop, connected to the flow from the loading pump. That runs via the column and out to the waste, so that's now just cleaning the pepsin column, while we are using the gradient pump to provide a gradient to elute the peptides from the trap column onto the analytical column towards the mass spectrometer for detection. This slide nicely showcases why the setup is using 3 valves and 2 pumps for fast and efficient sample preparation, for cleanup and elution with parallel column cleaning and equilibration. Let's have a closer look at the pump setup. We're using the ultimate NCS 3500 RS pump or module that actually combines the binary high pressure gradient pump shown here on the left and the ternary low pressure micro pump as a loading pump shown here on the right. Also, the column compartment is included in this one enclosure. However, this column compartment is not used in the HDX setup because the columns are all placed in the cooling chamber as part of the HDX PAL autosampler. For the MS data acquisition, I already mentioned that there are different platforms possible, such as the QExactive platform, the Orbitrap Fusion Tribrid, and also the hybrid platform is equally applicable. However, one needs to consider the newer instrument platforms are providing higher scan speed, and with that, more data points are obtained up to 40 Hertz on the most recent instruments, and that's beneficial for the analysis. Also, an additional benefit of the Tribrid platform is when an ETD source is available, that enables higher order structure elucidation with an increased spatial resolution. Generally, low resolution instruments such as ion traps are ruled out for this workflow because the detection of the isotopic pattern of higher charged peptides is really crucial for accurate data analysis. There's a strong point I would like to emphasize here. A big benefit of the Thermo Scientific HDX workflow setup is that the user can freely choose between quite a large number of different Orbitrap-based MS instruments, whether it's a QExactive Plus or an HFX or an Orbitrap Fusion. Any of these instruments can equally be used in the workflow, and that allows quite a lot of flexibility. For example, if a customer already has an MS instrument, he or she can simply add the HDX front end. Or if a customer has several instruments, the front end can be transferred from one instrument to the other, so it's really flexible. In principle, even an Exactive instrument could be used for the MS data acquisition because for acquiring the different time points after the labeling reaction, the method comprises only full MS data or scans. But for the entire experimental setup, there's one experiment or data point required that includes to run the sample through the entire setup once, but skipping the labeling step. So that would be time point 0. That can also be considered kind of a discovery experiment that serves for the identification of the peptides that are associated to the protein of interest. It's really important to assess that good sequence coverage can be assessed using that enzyme with the protein of interest. And for this discovery type experiment, MS AMOS capabilities for high confidence peptide identification is essential. That's why we suggest a hybrid-tribrid or Q-Exactive platform to be able to cover the entire workflow, including this discovery type experiment. Now a few details on the data analysis. There are different software packages on the market. For example, one is free for academic users. It's called HDX Workbench. But there are two packages that we recommend. On one hand, we have the BioPharma Finder software that can perform both the peptide identification as part of the discovery experiment I just mentioned, and also the HDX data analysis. It also includes an HDX model that simulates the Hydrogen-Deuterium Exchange and back exchange process at a single residue level. It provides also deuterium uptake plots for the individual peptides and also protection factor plots that are shown here in the picture. What's shown here is the comparison of two samples in the sequence residuals representation. This type of plot can be used to evaluate the protein's conformational property. HDExaminer is a third-party software package provided by Sierra Analytics, and it provides full capabilities to analyze all data, including ETD data and also even top-down data. It provides different results, visualization plots, for example, these deuterium uptake plots for the individual peptides and peptide plots, and also, for example, a heat map where we see the protein sequence listed here with color-coded bars representing the individual peptides that were detected. And the color coding indicates blue, very low or no exchange, hydrogen versus deuterium, yellow a little bit, and red, very strong exchange. There are also butterfly plots that are comparing the two different samples, indicating the degree of exchange based on the peptide number. Then we have deuterium uptake curves that also show the deuterium uptake of an individual peptide over time. And lastly, we have the view of the isotope patterns of individual peptides and how they are developing over time, or over the time course of the experiment. All these results can be directly mapped onto a protein sequence, a three-dimensional structure rather than a protein sequence only, but that's requiring a separate software called PyMole. It's a little software tool that reads all these results and maps them on a 3D structure. And one can nicely visualize different time points and see how the exchange in certain regions of the 3D structure develops over time. I think it's a very nice way to visualize. However, as I already mentioned, it requires that a three-dimensional structure for the protein of interest is available. Now, you may ask yourself, do we have any users out there already who are applying the Thermo Scientific HDX workflow? And the answer is yes, we actually have quite a number. And only some of them are listed here, showing or represented with the company logos. And I'm sure you'll recognize many of these names. But we also have a long list of academic customers that contribute strongly to the growing number of publications that's shown here on the plot on the right side. When we review the number of publications, taking all years together and compare users who have used Qtoph-based HDX workflows compared to Orbitrap-based HDX, the Qtoph numbers are a bit higher. However, if we just review the last, or consider the last 2 years, the Orbitrap-based HDX workflow users or publications are about twice as high. So that indicates a strongly growing number of papers based on a growing user base. So let's wrap up with a list of points and arguments why to choose the Thermo Scientific HDX LC-MS workflow. First of all, it's easy to use, it's future-proof, and it's really flexible. The flexibility refers to, on one hand, the advanced method development, but also to the option to hyphenate any Orbitrap-based mass spec to the HDX front end. The HDX Power autosampler has very low carryover, that's what it's really well known for, provides excellent reproducibility, can perform parallel equilibration. That's also made possible by the use of the 2 pumps and the 3-valve setup, which I've explained in detail. A single software control provided by the Leap Chronos software is integrating the entire sample preparation, the time scheduling, the LC and the MS control, and that ensures a really smooth run through the entire experimental sequence setup. The high resolution and accurate mass, the data that's delivered by the Orbitrap mass analyzer, is very well known around the globe. And lastly, the fast technical support that's provided, I believe, is also a very strong argument. So I hope this overview is helping you to better understand the scope and also the setup of the HDX workflow with all the hard and software modules involved. Thanks for watching.